All right. So welcome to our journal club. Um, I will be presenting this paper. USP1 contributes uh, to mitochondrial quality control in glioblastoma cells. So some introduction. So long non-coding RNAs or lncRNAs are a type of RNA with transcripts exceeding 200 nucleotides. They're fundamental regulators of transcription. They act as signals, decoys, scaffolds, and enhancers to regulate transcription and translation. And they play crucial roles in a variety of physiological processes, including cell proliferation, apoptosis, cell fate determination, chromatin modification, and RNA processing. Um, Dysregulation of lncRNAs has been implicated in various human diseases such as cancer, and lncRNAs are emerging as new factors in regulating mitochondrial function and mitophagy. And specifically in cardiomyocytes, lncRNA H19 inhibits excessive mitophagy by limiting PINK1 translation, therefore alleviating cardiac defects that occurs during obesity. So nuclear genome encoded lncRNA mallet1 binds to, oh, can you see my screen? Okay, there we go. Yeah, so nuclear genome encoded lncRNA mallet1 binds to mitochondrial DNA and functions as a mitochondrial epigenetic center to control metabolic reprogramming and mitophagy in hepatocellular carcinoma. It also regulates mitochondrial homeostasis and enhances the resistance of cardiac microcirculation to hypoxic injury via the mirror 26B 5P MF1 axes. So the lncRNA Shing14 is highly expressed in cerebral ischemia reperfusion injury and hypoxia reoxygenation induced neurons. It promotes oxygen glucose deprivation reoxygenation induced neuron injury by inducing excessive mitophagy via MIR1 182E5P bin P3 axes in the mouse hippocampal neuronal cells. So just some background on glioblastoma or GBM. It's the most serious type of cancer originating from astrocytes that occur in the CNS and spinal cord. It has high morbidity, mortality, and recurrence, and the median survival for patients is less than 2 years, even with treatment. And the current therapeutics are a combination of surgical, radio and chemotherapies. And autophagy induction is one of the hallmark effects of anti-glioma therapies. So targeting mitochondrial dynamics through autophagy might be a potential therapeutic strategy here. Here specifically, they identified that USP30-AS1, which is an antisense lncRNA located on the opposite strand of the USP30 locus, is upregulated in glioma, particularly in the most aggressive glioma, GBM. And high level of USP30-AS1 is associated with poor survival in both primary and recurrent glioma patients. And USP30-AS1 negatively regulates mitochondrial homeostasis and mitochondrial uncoupler-induced mitophagy in glioblastoma cells. And USP30-AS1 mediated repression of mitophagy may contribute to the loss of mitochondrial homeostasis and tumor development in glioma. So the first figure, they show the locus of the antisense part of USP30 in relation to the USP30 sense locus on chromosome 12. And then they analyzed the expression level of USP30-AS1 in lower grade or LGG glioma and high grade glioma using data from GEPIA or gene expression profiling interactive analysis. And they found that USP30-AS1 expression level was higher in both LGG and GBM tumor tissues when compared with normal tissues. High-grade GBM expressed more USP30-AS1 than the lower grade tumors. So overall survival and disease-free survival analysis are seen here in figure C. And that revealed that the LGG and GBM patients with higher levels of USP30-AS1 had poor survival outcomes. And then they explored the Chinese Glioma Genome Atlas, or CGGA, and that contained brain tumor data sets with more than 2,000 samples from Chinese cohort sets. And the high-grade glioma expressed remarkably higher amounts of USP30-AS1 than the LGG gliomas. So the high level of USP30-AS1 was associated with poor survival in all grade primary glioma, recurrent glioma, and high grade primary glioma patients, indicating that USP30-AS1 is a potential biomarker for prognosis. They then generated stable USP30-AS1 shRNA cell lines using a lentiviral vector. They generated 3 shRNAs and effectively knocked down the expression of the endogenous USP30-AS1. Immunoblotting revealed that USP30-AS1 all 3 shRNAs significantly reduced levels of inner and outer mitochondrial membrane proteins such as TIM23, TOM20, and COX4. The transient transfection of USP30-AS1 siRNAs showed this similar effect as well. So they then performed a quantitative RT-PCR to examine the transcript abundance of the mitochondrial protein. The mRNA levels of TIM23, TOM20, and COX4 were not affected by USP30-AS1 silencing, as you can see in the chart on figure D. The mitochondrial number can be predicted by quantifying the mitochondrial DNA copy number using RT-PCR. And they determined the contents of mitochondrial genes for tRNA and 16S RNA depletion of USP30-AS1 significantly reduced the mitochondrial DNA copies. So in the next figures, they overexpressed USP30-AS1 in U251 cells, and which is a glioblastoma line, and analyzed its effect on mitochondrial protein expression. Using qPCR and immunoblotting, they showed that overexpression of USP130-AS1 had no obvious effect on those biomarkers. These results, they indicated that USP30-AS1 may regulate mitochondrial homeostasis, although there was no significance on those. So mitophagy can be monitored biochemically as well through the degradation of mitochondrial membrane and matrix proteins. Here they used FCCP, a potent uncoupler of mitochondrial oxidative phosphorylation, to induce mitophagy, and their immunoblots revealed that depletion of endogenous USP30-AS1 by shRNA promoted FCCP-induced degradation of mitochondrial inner and outer membrane proteins without affecting the LC31 to LC32 transition. And the degradation of MF1 was also accelerated in USP30-AS1 depleted cells. So to demonstrate that mitochondrial proteins were degraded by autophagy, the cells were treated with a lysosome inhibitor, baflomycin A1. Baflomycin significantly reversed FCCP-induced degradation of TIM23 and TOM20 in both the control and the USP30-AS1 depleted cells, indicating that USP30-AS1 shRNAs promotes FCCP-induced mitophagy. They also transiently transfected U251 and A172 cell lines with USP30-AS1 siRNAs and analyzed its effect on CCCP-induced mitophagy. USP30 AS1 siRNA promoted CCCP-induced degradation of TIM23 and TOM20. As you can see. By contrast, overexpression of USP30-AS1 suppressed the FCCP-induced mitophagy. So the damaged mitochondria are selectively engulfed by autophagosomes and delivered to lysosomes for degradation during mitophagy. And they co-stained mitochondria and lysosomes with fluorescent dyes and examined their colocalization and control and depleted cells. And the silencing of USP30-AS1 increased mitochondrial lysosome colocalization and control and FCCP treated cells. So the mitochondria are primarily responsible for producing ATP via oxidative phosphorylation. They analyze the effect of USP30-AS1 on intracellular ATP levels. Depletion of USP30-AS1 remarkably reduced ATP in both the control and the treated cells. They then looked at ROS. So mitophagy can act as a mechanism to reduce mitochondria-derived ROS. Treatment of mitochondria uncoupler FCCP resulted in increase in ROS production. Silencing of USP30-AS1 decreased FCCP-induced mitochondrial ROS levels, which was significant according to their chart on the left. So finally, their evidence suggests that the mitochondrial-associated deubiquinase USP30 is a negative regulator of mitophagy. Therefore, they wondered whether if USP30-AS1 regulated mitophagy through modulating the expression of USP30. So they performed gene correlation analysis using mRNA-seq data from the CGGA database. The results revealed a negative correlation between USP30-AS1 and the sense gene USP30 in all primary grade glioma and grade 4 glioma. So they further looked at the effect of USP30-AS1 on USP30 gene expression in U251 glioblastoma cell line. The knockdown of endogenous AS1 by siRNA did not affect USP30 mRNA and protein expression. And overexpression of USP30-AS1 also showed no detectable effect on USP30 expression. Therefore, they proposed that AS1 does not regulate USP30 gene expression in a cis-regulatory manner. So they identified that lncRNA USP30-AS1 is upregulated in glioma and correlated with poor prognosis in both primary and recurrent glioma patients. They propose that USP30-AS1 mediated repression of mitophagy may contribute to the loss of mitochondrial homeostasis and tumor development in glioma. And lncRNAs are involved in the pathogenesis of various types of cancers, and alterations in lncRNAs expression contribute to tumorigenesis and metastasis. And numerous lncRNAs are expressed in tumor tissues or body fluids, making them effective biomarkers for early diagnosis and prediction of appropriate therapeutic responses. And evidence can indicate that antisense lncRNAs can act as regulators of their counterpart sense mRNA by modulating chromatin structures. USP30-AS1 is transcribed in the opposite direction to the sense protein coding gene USP30. And they assayed for a cis-regulatory function of USP30-AS1. However, silencing of AS1 did not influence the expression of USP30, suggesting that AS1 may not, in a cis-regulatory manner, impact USP30 expression. So it's most probably not involved with AS1 mediated suppression of mitophagy directly. And that's the end.